now. So welcome everyone. Thank you so much for joining the RUNX1 Research Program webinar series. For those of you who I have not yet met or we have not met, I am Katrine Erickson. I am the Executive Director of this research program. We are a nonprofit organization and we are hyper-focused on supporting patients with RUNX1 familial platelet disorder with predisposition to hematologic malignancies. We do this through raising awareness, like through programs like this. We do this through building a patient community. And of course, we do this through funding research. And our research funding is really focused on discovering interventions that would prevent the onset of blood cancers in RUNX1 FPD patients. That's our mission. We are fortunate to have wonderful funding partners in Alex's Lemonade Stand Foundation and the Leukemia Lymphoma Society. And just wanted to give that background to those of you who may be new to the RUNX1 research program. We will continue to host these webinar series. We've found them to be very effective, and lots of people have found them helpful, not only from the research perspective, but also from the patient perspective. Today in this webinar, we will have mostly scientists, but as I mentioned, we want to make sure that patients feel included and integrated in these discussions. So please do ask your questions, regardless of what perspective you're coming from. So we'll start with Dr. Brown's presentation, and then if you do have questions, I would appreciate it if you could write it in the Q&A icon. That way I can systematically go through the questions at the end of the presentation. Try to refrain from using the chat just because it can get a little hard to follow and distracting during the presentation. So without further ado, I'd like to introduce Dr. Anna Brown. She is a funded RRP investigator, and she's the head of the molecular oncology at SA Pathology at the University of South Australia. She's a geneticist by training and an expert on germline RUNX1 mutations. She and her team have collected the world's largest cohort of next generation sequencing data sets from RUNX1 FPD families, and has made that data accessible and available to the RUNX1 medical community on a database. And I'm sure she'll share with all of you a little bit more information about that and how to get access. We do hope that everyone can use that and leverage it. So today, you heard a little bit already, she's going to share work on understanding what are those common co-occurring mutations, somatic mutations that is, across patients who have leukemia versus patients without leukemia, so that we can begin to get an understanding of what are potentially those somatic mutations that are driving the transformation from a pre-leukemic state to a leukemic state. So with that, thank you, Dr. Brown, so much, and looking forward to hearing your presentation.
Thanks, Katrine. And just to introduce myself on where our group is from. So we're from Australia. We work in a town called Adelaide. So I do research, obviously, into blood cancer predisposition. I also run what's called a molecular pathology clinical laboratory where we do gene sequencing for patients who have cancer. So we do a lot of the gene sequencing clinically that would be relevant for these patients. And so basically that's what I do a lot of, looking at both inherited and somatic mutations. And that's what I'm going to be telling you about today. So I am part of a large research group which was founded by Hamish Scott, and I run projects with him and also with Chris Hahn, who's another senior scientist. So you can see from the picture here, we've got a lot of people that work in our team across different projects. I want to thank upfront our RUNX1 database team. We've got scientists and curators, Claire Homan and Sarah King-Smith, Dave Lawrence, who is our software developer, who's made the whole project possible from that point of view, and Mark Armstrong, who was also helping us with bioinformatic analysis of these data sets that I'm going to show you the results of today. Just to give you a bit more information about who we are and what we do, our research group actually runs a study called the Australian Familial Hematological Cancer Study, and it was established by Hamish Scott in 2004 officially, although he'd been working in this area prior to that. And at that time, RUNX1 was the only known leukemia predisposition gene. So basically, our centre acts as a major referral centre across Australia and New Zealand for all kinds of blood cancer predisposition, for research and diagnostics, and as well as working in RUNX1, we work across the whole spectrum of inherited blood cancers in both discovering genes and characterising gene mutations, and we've published, as I've listed here, you know, a whole range of different predisposition genes over time. So at the moment we've got over 200 families enrolled, and as there's more gene sequencing being done routinely, I mean, this recruitment of families and patients with mutations is just increasing over time. So I think it's an increasingly important area, and one that we've been in for quite a long time. So in terms of RUNX1, Hamish has been working in this area for a very long time, as I said, and so we've got various publications over time in the literature describing mutations in RUNX1 as far back as 2002, and following up with various studies trying to understand mutations and how they affect protein function, how they predispose to leukemia. And I guess I would direct you to a couple of recent resources if you did want to sort of dig in a bit more generally to this area, if you're perhaps a newcomer to this area. And we've got a couple of publications that have come out recently. One is I guess a general review published in Blood, available online, and that looks at, I guess, general features across 3 different predisposition disorders, including RUNX1. So there's good general information to familiarise yourself with the area. And additionally, we've just had published in Blood Advances is a study focused on RUNX1, where we've published clinical and genetic aspects of 10 families that we've been following, some of which we've been following for decades. So it's a very comprehensive look at types of mutations and clinical findings. And indeed, we published our first analysis of the somatic mutations in tumours from patients with germline RUNX1 mutations in this paper. So it was our data and looking at the literature, and in fact it was the nucleus that has formed the current project that's now expanded into our worldwide sort of database initiative, because we realised that we could start to find patterns in the data by aggregating it from investigators all over the world. And we've been very lucky to have a good working relationship with lots of researchers and clinicians in this area who've been really happy to share their data with us. So just to give you a bit of an overview again for people who might be newer to this area, so germline RUNX1 mutations cause familial platelet disorder with predisposition to myeloid malignancy. So if you look that up in the online inheritance database, that's what it will be called. So in terms of blood abnormalities, the main observable difference for patients who have a RUNX1 mutation is that they'll frequently have mild to moderate thrombocytopenia, and there are some potential defects in platelet function. We know that they have a 40% risk of developing a blood cancer by the age of 50 years. We know that myelodysplastic syndrome and AML are the most frequent blood cancers, but we also know that there are a subset of patients who develop lymphoid leukemias most commonly. They're more often pediatric cases, and they develop T subtype acute lymphoblastic leukemia. So the average age of onset of leukemia is around 30 years for patients who have a RUNX1 mutation, although we know that this actually spans the entire age spectrum. We have childhood cases, as I've just mentioned, but we also have people, you know, developing leukemia throughout their lifespan. And there will be some people who can get to, you know, quite older ages without developing leukemia. And I guess this family that we're showing on the right here that has a clearly damaging mutation in RUNX1 is a family that we've been following for a long time. And if we look at the patients who are carriers, so that with the pluses, they have the mutation, we can see all different ages of diagnosis in 50s and 60s. different types of leukemias, so acute lymphoblastic leukemias. Here we've got a pediatric acute lymphoblastic leukemia, and some patients who we're following who have yet to develop leukemia. And I guess this has been a mystery, and it's something that we really want to solve. It's that at an individual personal level, how can we better understand the factors that are going to contribute to someone developing leukemia? Because unless we can do that, we don't know how we can intervene to stop that process. We also know that there are other phenotypes associated with RUNX1 FPD. So we've characterised that some patients have very severe psoriasis and eczema and juvenile arthritis. So other things related to inflammatory phenotypes. And I think other people, for example, like Nancy Speck recently have been looking at those kind of aspects in their talks. And I think through the NIH natural history study, which we're going to hear about in the coming months, we're going to start understanding that the spectrum of phenotypes associated with these inherited mutations is much broader than had been previously annotated in the research literature. So that gives us more opportunities to try and understand how mutant RUNX1 affects cells and understand the predisposition to leukemia. So in terms of the different types of mutations, so this is taken out of our recent Blood Advances paper, where we, for the first time, collated all of the mutations that have been published over 20 years in the literature, trying to see whether there were specific patterns. And we found that there were 130 families that have been published since the first description in 1999. And what we found was there are all different types of mutations that affect RUNX1. So on the top, we can see that there are frame and stop gain mutations that would likely truncate the protein and remove function of that allele as the most obvious mechanism. On the bottom, we've mapped the missense and the splicing mutations, where we can see clustering of those in the RUNT domain, and we know some of those can affect protein function through disrupting DNA binding to protein partners, exclusion from the nucleus. And then there are a range of partial to whole gene deletions. So as a general mode, we're looking at like a reduced expression or reduced function, I should say, of the RUNX1 allele being the first predisposing event. So as part of our process of creating the database, we actually talked to everybody who'd published in RUNX1. We talked to colleagues that we knew were working in this area who might have families that they hadn't published. And what we found when we did our first survey, and we're about to start updating that survey again this year, so this is from 2 years ago now, we found that there are actually over 200 families that we've annotated across 52 centres around the world. So we've got an increase immediately from 130 to 200. And then we could start looking at ascertainment by the number of families per population. And in Australia, I guess because it's a small population, we've been working in a long area, we had the highest rate of ascertainment. So if we extrapolated that to worldwide, we would think that there are probably thousands of families that exist that haven't been discovered. So we've probably got a fairly low ascertainment rate. So I think that means that one of the significant progress in the field is still going to be correct and timely identification of families. And that's kind of where our database is now using this information to turn, as part of our data set, into a mutation registry. And this is actually the front page of our database, and basically anybody can email me and get a login to look in here. And this part is under construction, but what this shows are the known germline mutations in RUNX1, and the idea is to have an interactive interface where we annotate the information. People can go in there and they can look at more information for all the variants that have been pulled out of lots of different databases, cancer databases, population databases. They can look at whether the mutation is predicted to damage the protein. And importantly, we're also looking at retrospectively going back through the literature, which we've done, but updating the clinical classifications for those mutations. So since 2015, there have been classifications that are defined by the American College of Medical Genetics, and last year we had a committee that defined special rules for RUNX1 mutations. So that can be really important in telling you, if you find a mutation in RUNX1, whether that is a previously seen mutation, whether it's a clinically valid mutation, because that's important information for the patient and the doctor in terms of trying to figure out how to manage their risk for leukemia. And importantly, will also be linked directly to entries in the clinical genetics database, which is called ClinVar. And so we encourage people to come to us with their variants. If they've got some, we're happy to help them. We're happy to put them in the database. People can put their own classifications in the database as well. So while it's kind of curated by us, it's also interactive by other users. So in terms of looking at somatic mutation data, while we were doing a survey, or at the same time we were doing the survey of the families, we were also asking investigators about what data sets they may have that may or may not be published. And we managed to collect around 170 data sets that were next generation sequencing, so gene panels, or whole exome sequencing of samples from patients who are RUNX1 germline carriers. So we got data from 19 different groups, and you can see the data sets range from 30 data sets from some groups who've been specialised in that area, down to single data sets from patients who might see patients in the clinic and have some clinical sequencing, but that's not a research specialty. So we collected data from everywhere we could. And that represented 62 families and 111 different individuals with RUNX1 mutations. So this is just a breakdown of the samples. So we collected about 60 data sets from sequencing of tumours from patients with germline RUNX1 mutations, as well as around the same number of blood samples sequenced from patients who had not developed leukemia. And we can see, if we look at the breakdown of malignancy, by far the most common leukemia types are MDS and AML, which is what we already know. So just going over what we did, I don't want to go over this in detail, but the information is available if people are interested. What we did was collect raw gene sequencing data from NGS output from people from the groups wherever we could, and designed a pipeline that was able to take data of any type. So as you can imagine, these people had generated this data at all different time points. Over the years, and technology changes a lot during that time. So we had exomes and myeloid panels and custom panels and different technologies. And so we've designed a pipeline that can take all of that data and try and treat it in as most consistent a way as possible, so that we could have a standardized, as much as we could, data set for filtering and curation to look at the mutations. And to do that, basically, we've got custom designed software called Variant Grid that helps us do automated data filtering. And again, this is the front page. Our website down below, where our registry diagram is, we've actually got an oncoplot that's starting to, or does show the output of the tumour sequencing data that we filtered within the database using this software. And in fact, that's interactive, an interactive portal into the database. So in terms of how that software works, it can automate a lot of the filtering things that we would normally do. So in terms of in a research setting, and also in a clinical setting in my lab, if we're doing clinical sequencing, we have a number of parameters that we commonly use, and that are also defined by various societies and Association of Molecular Pathology, for example, that use different metrics to try and look at somatic mutations in data sets where you don't have a matched tumour and normal control. So we have to remember that when we've sequenced the tumour here, we've actually sequenced all of the inherited mutations as well, not just the somatic mutations. So without that direct control to subtract this inherited mutations, we need to use parameters such as the presence or absence in normal population databases. We need to use things to look at whether that mutation is predicted to be damaging, so that we can pick the highly damaging mutations that are more likely to be leukemia associated. We can look at the genes specifically by looking at genes we know are involved with RUNX1, genes that we know are involved in leukemias already, and cancer databases such as the Cosmic Cancer database, where we can cast the net more widely in order to perhaps identify genes that we hadn't really looked at before in this context. So we did all of those things, and this can be automated in our software, and it's fully customizable. So people with logins who have their own specific gene that they want to look at, or specific parameters that they're really interested in looking at in the database, can go in and make their own custom analyses. But we also have preloaded analyses based on what I've described here as well, for people who just want to look at the actual output in a particular tumour, for example, of what we found. And after we did that filtering step, we've also got a manual curation step, which is looking at the raw data, making sure they're real mutations and not artifacts. And then for some of the mutations that maybe are in new genes that we haven't looked at before, we'll look a bit more closely at them and apply other criteria to see whether we think there could be new candidate leukemia genes or not. So just to show you, I gave a webinar late last year looking at the somatic mutation output of tumours from germline RUNX1 carriers, and this is just a summary slide, I guess, of the most commonly mutated genes. So we found actually 97 genes that had mutations that passed all of our filtering and curation. The majority of those genes we saw only mutated once in one tumour, so it's really hard to draw conclusions about their general involvement. And I guess more data may help us build up additional cases for some of those other genes, and thereby understand new genes that are involved in that process. But I guess in terms of the genes that we already knew to be important in development of acute myeloid leukemia and myelodysplastic syndrome, we could easily look at some of these genes and find patterns. So here we've got the gene names ranked by how frequently they were mutated in tumours. And what we found was that a second mutation of RUNX1, so a somatic mutation of RUNX1, was definitely the most common somatic finding in tumours of patients with germline RUNX1. So this is not an unusual scenario for a gene that you would broadly classify as a tumour suppressor, that you would get mutations in both alleles, increasing the leukemic effect of that gene, and that's in fact what we've seen here. Interestingly, we saw mutations in genes called PHF6 and BCOR that were also frequently mutated, and also recurrent mutations, you know, in a range of other genes such as TEP2 and WT1, and then going down in frequency from there. So these are genes that are known to be mutated in myeloid diseases, but what we found from our analysis is that compared to leukemias that have a somatic RUNX1 mutation, the other genes that are mutated with RUNX1 are different if you've inherited your RUNX1 mutation. And that's what this is showing here, is that if you have an inherited mutation, you're far more likely to get a second RUNX1 mutation in your leukemia than if you're a patient who just develops a single somatic RUNX1 mutation. And PHF6 is also, you're far more likely to get a mutation in that gene if you've got an inherited leukemia. And so that's telling us that there is something qualitatively or biologically different about a predisposition disorder with RUNX1 in terms of the genes that are important for development of leukemia. And that is important information to know, to direct researchers specifically, because if people are you know, in the general field of myeloid malignancy focusing on the most common genes, they might not be looking at the genes that are the most important ones for patients who have a predisposition disorder. So what we wanted to do next was move on to our second data set, which was to look at whether there were mutations detectable in the blood of patients who didn't have leukemia. So there have been studies published in the past that have started looking at this. So Lucy Godley's group at the University of Chicago published a study in Blood in 2015, and this figure on the left is from their paper. And basically looking at carriers in RUNX1, which are these patients here with the green bars who didn't have leukemia, and at different ages, they found that they could see an increase in somatic mutations the older that patients got. And that was a general increase in a number of mutations that they could detect in the blood. And in one case, they found a known leukemia-associated mutation. We and others have also in the last few years published examples of mutations found before leukemia. So we know that we've seen, single cases or a couple of cases of mutations in genes such as DNMT3A, SRSF2, TEP2, KRAS and BCOR. I think I presented at the end of my last seminar where we just started looking at the pre-leukemic data that, we could see that there were going to be things going on, because upon reanalyzing this particular sample where a patient has this germline RUNX1 mutation present at 50% mutation load, which you would expect, when we carefully reanalyzed their blood data, we actually found 5 different somatic mutations in DNMT3A, BCOR and BCOR-L1 that we could see at a lower level. So I think, knowing which genes may be important in the tumours then also lets you go back and look in more detail in the pre-leukemic samples as well. So I think, so we knew that there were going to be mutations, but we didn't know how common. So that was really how common are those mutations, which genes did they most commonly occur in, and what are the ramifications of having this information. So just to remind you, we had 65 samples from pre-leukemic carriers, and this represented 56 individuals from 33 families with 28 different germline RUNX1 variants. We had a fairly even break down between males and females, which is good representation. And we also had a really broad representation of age ranges. So we had people from 1 year of age to 76 years of age with sequencing of their blood, and none of these patients at the time they'd been sequenced had developed leukemia. So it's quite, it's a good sampling of the different ranges of this predisposition disorder to try and get a handle on what's going on in the blood of patients before they get mutations. So what do we find? So out of the 56 individual people, so some people we had multiple samples, and I'll show you some of that data later, we found that 20 patients had what we would classify using the techniques I showed you previously, clinically relevant somatic mutations. So somatic mutations that we could recognize in genes that are known to be associated with leukemia development, and mutations that are predicted to be damaging to those genes in the way we know that they can cause leukemia development. So that is a little over a third of these patients. The people that were unselected, apart from having a RUNX1 germline mutation, you can just sequence their blood at any age. And in a third of them, you'll find somatic mutations. So this was, I guess, data we just didn't know, and it was potentially more common than I would have been predicting. But what it tells us is that in patients where, it's not routine to do molecular monitoring on blood of carriers, we're missing a lot of information that potentially is there and could be quantified. So in terms of the genes that we found, interestingly, we found the most common gene that we found mutations in pre-leukemic carriers was a gene called BCOR. And also the other 2 most common were genes called TEP2 and DNMT3A. And because it's still a small sample size, there is quite a long tail of genes where we found single gene mutations. And again, as we accumulate more data, we may see some of those percentages shift up and find a greater number of genes that are important. But I think we can see from the number of mutations that we've got so far, which we can show on the right here, individual mutations, we can see clearly that BCOR, TEP2 and DNMT3A are going to be important pre-leukemic gene mutation targets. So what I've plotted on the right-hand graph is the VAF, otherwise known as the mutation load. Our lower cutoff for detecting mutations above background was 3%, so we would look at anything that was 3% or higher. And we can see that we can get a range of mutations in these carriers from that low 3% all the way up to 40%. So we had people who had what looked like a somatic leukemia-associated mutation present at quite a high mutation load without getting leukemia. And obviously, we're going through the process of following up on these patients and these data and confirming that, in fact, all of those patients, when they had collected the sample, didn't have leukemia. Because sometimes your clinical ascertainment of a patient can change over time. But at the information we have at this point, we can have patients with quite high mutation load and still not develop leukemia. We've been looking at whether the type of germline mutation that you inherit changes whether you can see mutations in your blood before you get leukemia, because potentially this is a specific mechanism that is more frequent in patients with a particular type of disrupted function of RUNX1. And if we look at these mutations mapped onto the RUNX1 protein, again, we can again see that there doesn't appear to be much difference in patients who we didn't find a somatic mutation, which are mapped on the top, and the patients where we did, we can see all different types of mutations represented. So as far as we can tell, this is a common or occurrence across all of the known predisposition mutation types. We also have been looking at age. So if you just look at the average age, which is this graph on the left here of all of the samples and those versus no somatic mutation detected versus there were somatic mutations detected, there's no significant difference, although it does look like the average age, if you have a somatic mutation, is slightly higher. And if we break down the data based on individual pool rank by age, as we can see on the right-hand side, and mark the people who we did find mutations in red compared to blue, we can actually see patterns emerging. And so what we can see is that nobody under the age of 16 years had mutations detected in their blood, and everybody over the age of 70 years, remembering that the small numbers, still had a mutation. So that suggests that you're getting an increase in your mutation load as you age. And this is consistent with a general mechanism that we also observe in healthy individuals called clonal hematopoiesis, where even patients that don't have a predisposing mutation collect mutations in their blood as they age. And some of those mutations are leukemia-associated mutations that do, in fact, increase their risk of development of leukemia. So it's a question of, how much that process looks like the process that we're seeing here in RUNX1 carriers. What we also found when we looked more closely was that older individuals had more mutations per sample. So if we look on the right on the bar graph, we can see apart from one outlier sample, everybody up to the age of 50 years, we only observed one mutation in their blood. Everybody who was over the age of 50 years had 2 or more mutations in their blood. And we can plot these gene mutations out on the left by patient, showing you the gene name and the mutation load. And apart from our single outlier, we can see that, generally patients who are younger have one mutation and their mutation load is quite low. However, as they age, they tend to acquire second mutation, and they have a variable range of mutation loads, which suggests maybe we're getting independent mutations in different cells arising in the same gene, because you can see often where we can get mutations in 2 mutations in the same gene, like B core, so we don't know if they're in the same cell or different cells. Sometimes we'll get different combinations of those common mutated genes mutated together. So patients who have a TET2 and a DNMT3A mutation together, for example, we've seen multiple times B core and DNMT3A mutated together. So we think that there's a dynamic and ongoing accumulation of these kind of things as you age. What was important for us to know is whether these pre-leukemic mutations, how they relate to the mutations we see in tumours. So are we finding mutations in the same genes, Or are they a different set of genes? How is that related to that process? Because what we really want to do is to find a process that we can measure that's directly related to the risk of developing leukemia. And what we found was that all of the common pre-leukemic somatic mutations that we found, so in the orange bars here we found, you know, B-core, DNMT3A, TET2, they're all mutations that we do find in RUNX1 germline tumours. So you could infer from that that some of these clones truly are pre-leukemic clones, and in some cases can give rise to tumours because they have the same or similar mutations. However, if we compare the frequency of somatic mutations in tumours in blue compared to the pre-leukemic samples in orange, we can see that while there's overlap with the genes, there are also differences in the genes. So for example, somatic or a second RUNX1 mutation in tumours, which we see most frequently in tumours, we have never seen that in a pre-leukemic sample. And PHF6 is also far more frequently mutated in end-stage leukemic samples than it is in pre-leukemic state. So what we infer from that is quite probably that somatic RUNX1 mutations may be a late event in the leukemic process, and that they may occur commonly across some of the different types of pre-leukemic states. And in fact, although I haven't got the specific analysis to show you for this yet, you can see somatic RUNX1 mutations occurring with these pre-leukemic mutations. So they occur with B-core mutations, and they occur with TET2 mutations. So suggesting you might set up a pre-leukemic state and then attract or develop rather a somatic RUNX1 mutation later in the process. So what do we want to use this data for? We want to use this data for multiple things. We want to see how we can use this data to tell us about the individual risk for a patient who develops these mutations, for one thing. And in the field of non-predisposed leukemia, people have been looking at this for, you know, a number of years since clonal hematopoiesis was discovered about 7 years ago. In papers that were published a couple of years ago and continuing, people have been trying to look at the risk of these mutations. So we can look at what's been done in that field. And here we've got a summary of a figure from a paper published in Nature a couple of years ago, where they did a lot of retrospective sequencing of people who were at the time healthy individuals. And then they followed them, and they've defined which people over a time period didn't get leukemia and those that did. So they defined the people who didn't get leukemia as controls, and the patients who did get leukemia as pre-AMLs. So they had 676 controls, 124 people who went on to get AML. And then they looked at what gene mutations they had in their blood. They looked at what mutation loads were. They looked at the frequency of those mutations. And we can actually have a look at that data to look at what are called the hazard ratios, which is what is plotted here. And so looking at their data, they developed a hazard ratio which tells you over a 10-year period, if you have a mutation in a particular gene, what your increased risk is in developing AML. And so we can see here, looking at these green circles, what the hazard ratio is for various genes. A hazard ratio of 1 is no change, and obviously the more that it increases, the greater your chance of getting leukemia. And in fact, some of the same genes that we have seen in our RUNX1 samples are also represented in these populations, and so that can start to give us potentially an idea. And so we can see that out of the commonly mutated genes that we've seen in RUNX1, B-core, DNMT3A, TET2, and actually we had a few SRSF2 cases as well. Out of those genes, B-core actually had the highest hazard ratio. So in their data set, B-core mutations were associated with the highest risk of progressing to AML within the next 10 years if that mutation load was also changing over time. But what we can also see if we look at the column here that's called frequency is that these dots are really small compared to something like DNMT3A, and that tells us that in a otherwise healthy individual with no predisposition mutation, they don't very often get mutations in B-core. So we thought this is worth looking at more closely, like what is the difference in the genes that are mutated in a pre-leukemic state in someone who has a RUNX1 mutation versus someone who doesn't. So we've taken data that was available from another one of these big clonal hematopoiesis studies and just plotted the frequencies with which different genes are mutated or preferred in these populations. So compared to the control population from the clonal hematopoiesis study in the blue bars, we can see that DNMT3A is by far the most commonly mutated genes, and BCOR is really a very small histogram bar indeed. So it occurs, but not very commonly. In contrast, if we look at our RUNX1 pre-leukemic carriers, we know that BCOR is far and away more common compared to a general population, and indeed we have also fewer DNMT3A and more TET2. So that suggests that there's a different profile or a different selection for different gene mutations if you have a germline RUNX1 mutation compared to if you don't. And again, this is important information because we don't, if you're doing general research in the area targeting pre-leukemic mutations, you don't want to rely on the general clonal hematopoiesis community. You want to define for the specific population of patients that you're interested in helping what's important for them. We also had a look at this data, bearing in mind for all of these comparisons that our numbers are still small compared to doing large population studies, at the accumulation or the frequency or the likelihood that you will have to have a somatic mutation in your blood as you age. So while in orange we know that the risk of getting somatic mutations in the general population increases with age, overall as a percentage of the population, it still is quite low. Whereas we know from looking at the data we've looked at already that in RUNX1 carriers, your chance of getting mutations increased over time significantly, where patients over 60, small numbers, but all of them actually had mutations. So we'll be interested to see whether, when we collect more data, whether that is maintained. And it kind of suggests that if we're seeing clonal hematopoiesis, it's a different type of clonal hematopoiesis. It's different genes that are important, and it's a more exaggerated effect in terms of how many people it affects. And I guess even while we don't know the mechanistic processes that cause those differences, it does give us a potential molecular explanation for why there is an increased risk of leukemia in patients who have inherited RUNX1 mutations. So in the last part of this talk, I want to take you through a few case studies for patients where we had multiple samples. So we can do population estimates and we can look at the data in the way that I've shown you. But in terms of if we want to build risk estimates that we can apply to, I guess, individual people, if we are monitoring their blood with gene sequencing, we really need to have serial samples over time. And I think we're going to hear a lot more about this kind of information with the NIH natural history study that's going along prospectively, but also we can do retrospective studies. So for example, we're doing follow-up information on all of the patients that we've sequenced at the moment. Some of those samples in those patients will have been collected years ago, and we'll be able to know compared to what the mutation state of their blood was years ago and their clinical state now, we'll be able to start building a risk profile similar to what's been published for normal populations. So we have a few case studies where we did have multiple samples. So this first one is from a family that has a RUNX1 R320 stop gain mutation. They came to our research study because Dad was diagnosed with myelodysplastic syndrome, and at the time I was also recognised that his daughter had thrombocytopenia. Unfortunately, his MDS was quite aggressive and he passed away soon afterwards. When we did gene sequencing of his tumour cells, we found in addition to his germline RUNX1, he had somatic RUNX1 mutation, which I've shown you is a classic manifestation of the disease, but we only see it associated in patients who already have leukemia, so that's consistent with what we know. And since that time, that the family came to us, we have been collecting blood samples from the daughter who at the moment has thrombocytopenia only. So we've actually been collecting samples from when she was first diagnosed at the age of 33, and she is now in her early 40s. And what we can see from doing the sensitive gene sequencing in our lab and analysing it the way I've shown you was that at 33 years of age, we could detect a damaging mutation in TET2 at quite a low mutation load. However, in subsequent samples, we can see now that this mutation is starting to increase in its mutation load, and it's now at 5%. So this matches with what we've seen with the other data. She's still quite young, so we've still only seen one mutation, and we're seeing one mutation at quite a low mutation load. So that fits the profile that I've shown you overall. But what this tells us that the other data can't tell us is that we know that at the last time we checked in with her, she's about age 42 now, she's still well. So we know that a mutation that we could detect at 33 years of age, 9 years later, still has not led to leukemia. So that starts to be able to, for us to be able to tell what are the timeframes that we are looking at in terms of first detecting mutations in patients at particular mutation loads and the time to when they might be at risk, higher risk of developing leukemia. This second case study is looking at a family with a missense mutation in the RUNX domain of RUNX1. So there are 4 individuals here who are carriers, 2 of whom have gone on to get AML, 2 of whom have thrombocytopenia, and at last follow-up last year were otherwise healthy. We again sequenced the AML tumour cells from one of the individuals who developed leukemia, and again we identified a somatic RUNX1 mutation that we commonly see but only at the leukemic stage. This individual here on the left, who's actually been a bone marrow donor for one of his siblings, they're currently all well at the moment, which is great. We were able to do sequencing at a couple of different time points. And at the age of 43 years, when he presented and gave us a blood sample, we found a damaging mutation in DNMT3A, which is one of the common genes we found across the whole cohort. And again, we can see that in a 5-year period, that mutation is still detectable 5 years later in his blood, and has increased again to 5%. So again, he's within that age range where we've shown patients have one mutation at fairly low mutation load, but it's been another 5 years, I think, since we were last able to sequence a sample. So while we know he's still well, so there can be, you know, a potentially decades, over 10-year gap, we don't know what his blood looks like now and whether he's passed into another stage based on the other data we've collected, that over 50 years of age you start to see further mutations developing and mutation loads increasing. And the final case study is looking at a patient where we actually managed to catch the transition from some thrombocytopenia to leukemia. So this patient presenting consistent with what I've shown you before, the first sample we have from her, she's 54 years of age. She already has multiple pre-leukemic mutations. So in blue, at quite high mutation loads. So in blue, she had a damaging mutation in DNMT3A. She also had a mutation in TET2. And also in SRSF2. So there are 3 of the common genes that we've shown, and they're occurring together in this patient who's over 50 at quite high mutation load. And what is interesting here is that the next time point at 55 years of age, we can still detect those same mutations. Not much change in 2 of them, but DNMT3A increasing still. And then at age 56, so less than a year later, she develops AML. And when we see the gene sequencing on this, we see something different. So we can see the mutations that were present there before leukemia, but now what we've seen is a rapid acquisition of a somatic mutation in STAG2. So another known leukemia-associated gene. And we can also see a somatic RUNX1 mutation starting to appear as well. So I guess that fits with our hypotheses that RUNX1 mutations are later events in the process. And in this case, it may not be the main driving later event since we've got a STAG2 mutation, but worryingly, it also tells us what we suspected is perhaps that last mutational event actually precipitates quite a rapid progression to malignancy. And so when you're thinking about, you know, how you might look at this disease process, that will educate you, I guess, in terms of, you know, where you want to target your intervention. So if we can, I guess, summarize now at the end of what I'm going to show you today, what we think we know from the data we've looked at, we can build, I guess, an age timeline based on the data from the pre-leukemic patients that, you know, that says that up until the age of 15 years, you're not likely to see too much going on in your blood in terms of detecting mutation. Over that 15 years and up until the age of 50, patients may develop a single mutation that has a low mutation load, commonly in BCOR, TET2 and DNMT3A. And these mutations we know can persist for 7 years, over 10 years, potentially a long time without necessarily changing a lot. And once patients reach the age of 50, we're seeing an increase in the number of mutations, an increase in the mutation load, and presumably an increase in the risk of developing leukemia. And most worryingly, you know, we have an idea now that we think somatic mutations in RUNX1 and some of these other genes might be events that really drive that acquisition of leukemia, and that might happen quite fast. And so your opportunities for intervening when you see those mutations might be quite limited. So I guess in terms of that, you know, would it be better, or what we should be aiming to do is figuring out that if indeed these pre-leukemic clones are the seeds for the later leukemia, while we've got that time where we can see them in the blood, that's where we potentially be looking at aiming interventions. So this is a timeline based on pre-leukemic patients. So we know this also isn't accurate, because what we also know from the literature, and just to remind people because we've been so in the pre-leukemic space during this talk, is that there are patients along this timeline all the time who are developing leukemia at different ages. So out of all of the patients that develop leukemia, we know that there's pretty equal distribution in those different age ranges in terms of your risk of getting leukemia. So we know that in those cases, this timeline's not applying to them. So how is that different? Has that accelerated the timeline somehow been put into a small one? You know, are there other factors? Are there mutations in particular genes that are more common in the earlier timeframes? And so there's a lot more work that we can probably do, we definitely need to do to try and define that better. And as I was saying, the only way that we're going to be able to do that is actually by starting to do more molecular monitoring and looking at the mutations. And I guess what this study has also shown is that we have specific gene mutation target now from this data that we think are specific for germline RUNX1 carriers, where we can start designing interventions in this early phase rather than waiting to this more accelerated phase. So this is the last slide of my talk, just to reiterate what we think we found from this data, and I'm really happy to take questions in a minute. So we found that somatic clinically relevant mutations were detected in 36% of germline RUNX1 carriers without malignancy. BCOR, TET2 and DNMT3A were the most frequently mutated genes. We can see that single low mutation load or low VAF mutations can persist for years in carriers without developing leukemia. And we also know that the number of mutations and the mutation load increases with age. So we know that BCOR, TET2 and DNMT3A are also mutated in the tumours of germline RUNX1 carriers, making them therapeutic targets for pre-leukemic intervention. And we also saw very clearly that somatic RUNX1 mutations are not observed in the pre-leukemic state, and we think that they're, you know, associated with a later accelerated progression to leukemia. And that somatic pre-leukemic mutations in RUNX1 carriers, the proportions of genes that are affected are different from the normal age-related clone of hematopoiesis. So we really need to design our therapy development and monitoring specifically for these carriers. And as I said before, we really can only do this by collecting data, by directly monitoring. And now that we have some gene targets, we can start developing assays and models in the lab to try and find targeted therapies that will hopefully attack the cells that have those mutations and clear out those pre-leukemic clones in carriers before they can develop leukemia. So just finally, I just wanted to make sure, as well as acknowledging our research group, that we acknowledge the other departments that help us with our study. And of course, all of the wonderful international collaborators, which I've listed on the right here, who really generously contributed their data, which you can see already is allowing us to make observations that just weren't possible before. So thanks for listening, and I'm really happy to take questions.
Anna, that was a beautiful presentation and so insightful. So many takeaways. Thank you so much. I wish we had much more time for Q&A. There's lots of questions, and perhaps we can take some of these questions offline via email. But let's try to. I'll do rapid fire if we can. One of the common themes and questions is about the pediatric phenomenon. You know, why in your data set were you seeing so few at younger ages? So one question is that, you know, interesting that there was no one under 16 that had any detectable somatic mutations. Nevertheless, we know that there are children who under the age of 16 get malignant progression. Do you think that in those cases there's a big bang of mutations all at once rather than a gradual accumulation? And, or is there just no correlation between chip type mutations and progression? And this person talks about the Big Bang theory of AML, and that Tim Lay had considered this a while back, but didn't have much evidence in the early AML sequencing studies.
Yeah, that's a good question. I guess it's hard to answer without data. I guess the information that we've got at the moment suggests a kind of a gradual increase, at least in pre-leukemic mutations over time, rather than a big bang. But then I guess you could also see that what we haven't really been able to capture, except in one sample, is that transition from that pre-leukemic state to overt leukemia. And that may well be associated with a rapid accumulation of multiple mutations. So for example, in that one patient, if they've got a somatic RUNX1 and a somatic STAG2, and of course they could have somatic mutations in other genes that aren't drivers but still part of genomic instability. So that could essentially be considered a big bang that sets off that development of leukemia. But I guess the question still remains whether you need, you know, those other clones that occurred before have to be there first as well to collaborate. And I guess we don't know. The other thing I would say, for children, it's more often acute lymphoblastic leukemia T subtype. Maybe we're not looking in the right genes yet, because the tumours we've had to look at so far are quite small in number. We might not recognise what are the important genes for those, and if we, you know, we can get bigger studies of those, we might be able to find something that we'd kind of overlooked before, because we have been so much more familiar with pre-leukemic states for a myeloid disease, and we don't really know much about that in lymphoid disease.
Yeah, most of the panels that you mentioned were actually myeloid-based panels, correct?
Yeah, we do have a good number of exome data, and we try to be quite broad and look at new genes. But so I think, you know, that's where just having more numbers of samples might help some of those insights fall out.
Another question is, did somatic mutations run in families? In other words, did you see patterns within a family?
No, we haven't. So I'm just sort of thinking back through the tumor data, or the, you know, whatever data we have. So there've been a couple of studies in the literature where they suggested that specific somatic mutations could be recurrent in families. And one of them was explained by co-inheritance of, you know, a JAK2 haplotype, making you more likely to get a somatic JAK2 mutation in your tumor. We haven't actually seen anything similar yet, but I think one of the studies that we're going back to look at now is not just the somatic mutations, but what other germline mutations might be co-inherited with the RUNX1 mutation that might be influencing what they get in their tumor. So I guess the commonality is at the gene level at the moment, in that we know lots of people get second RUNX1 mutations, but the type of mutation they get is not the same. It can be all different kinds. So, yeah.
Another question that we received is, have you ever observed in FPD case studies a decline in VAF of any pre-leukemic mutation unexplained by expansion of a new clone with a second or subsequent pre-leukemic mutation?
That's a really good question, and I guess it builds on observations in clonal hematopoiesis that where they've got enough data that where they can say that certain mutations are more drivers than others. Some of them might just occur what you would call stochastically, so they occur by chance and they're in relevant genes, and you might get a bit of clonal sort of drift, sometimes even an expansion without really true driven selection. And in those cases, in normal people, you can see that clones will sometimes go down over time as well because they're not truly being selected for. We haven't seen that yet. It's not to say that we won't see it. I think it's just that we have a lack of serial samples over time in the same person to be able to tell whether that same phenomenon occurs. But it's important. It's important to know whether there are differences in particular mutations or in particular genes that are mutated that drive that advancement of that clone more strongly, because that will tell you about the risk for that patient.
Here's a question coming from a patient. Are there external symptoms to be aware of when someone develops mutations in those 3 genes that you described, for example, BCOR, TET2, and DMT3A? Or does the person need to have genetic work done in order to have a timely intervention?
That's a fantastic question. Again, I think we can go, from where we've looked at clonal hematopoiesis, so these mutations in the healthy population. So really the definition of clonal hematopoiesis is the presence of these mutations in the blood of healthy individuals. So I guess that's why it was undiscovered for a long time. They don't otherwise have abnormal blood counts. They're not unhealthy. They don't have any other symptoms, yet they have these mutations in their blood. So while I can't answer that question for RUNX1 carriers specifically, I think we're not aware of anything that correlates with the acquisition of those mutations. And in some ways, they're kind of a silent process that's going on inside the cell, and really the only way is to directly gene sequence to look and see if they're there, I think.
There's a couple of questions regarding the healthy RUNX1 carrier donor and whether or not that donor's mutation was observed in the stem cell recipient.
Another good question. I don't think we had the relevant sample for that patient, so I can't answer that.
Okay.
But yeah, certainly. So in terms of what we've seen in other predisposition disorders, we have and others have definitely observed unfortunate cases where patients who have, for example, a somatic DNMT3A mutation, and then they donated that stem cell to a family member, then the family member developed leukemia with that same somatic mutation. So it's definitely, a clinical outcome that their hematologist may want to take into consideration for how that family is going and monitoring them. At the moment, they're all well, which is great to hear. We always like to hear that.
Oh, that's great. Another question. I know we're a little over, but people seem to be staying. So if you're okay for a few more minutes. Yeah. Another question is, how often do you suggest, based on the data you presented today, to check for somatic mutations in the blood? And do you suggest looking at the blood or the bone marrow, or is the bone marrow preferred?
Yeah, really good questions. I think one of the reasons we really wanted to do more of this, in a research study, as well as because we don't have good evidence that we could develop a clinical guideline. So we actually really want to expand, we're looking at applying for grants now to really expand some of these studies to finding as many patients, enrolling as many patients in research studies so we can define that, because I don't think we're at the level where we could make a specific clinical recommendation. We don't have enough data. In terms of, although we have some hematologists who, as part of their monitoring, of the patient. So the patient might periodically go in to get marrow done or get their bloods done and have specific appointments. And I think, you know, that is still quite dependent on the particular relationship. But say if you go in, you know, every 6 months, every 12 months to see your hematologist, you know, at that time you could easily have gene sequencing done at those appointments on the bloods that are taken. I don't think, from what we know about mutation loads of leukemia-associated mutations, generally they are pretty well detectable in blood. So you wouldn't necessarily need to have a marrow done every time if you're just kind of looking to see if anything's changed at the gene level.
Great. Another question that came through is regarding the status of non-hematopoietic malignancies. Do you see that as a common trend across families?
So I think this is a point which we have, as a field, have not done a lot of detailed data collection. I think people tend to be really focused on the blood, you know, the platelet dysfunction and the leukemias because that's what they know about and that's what catches their attention. So I don't think there's any good associations. There aren't any good associations published, and even within our families, I don't think that we've collected good data that suggests that there is a strong association between another cancer type. But I'm not saying that there won't be, I'm saying that I think there's data missing there.
Regarding some of these these somatic mutations that are common in clonal hematopoiesis of indeterminate potential, are you also collecting phenotypic data on inflammatory states like cardiovascular outcomes, etc.?
Yep, good question. We haven't done that yet, but as part of our follow-up, we certainly can start asking those questions. And our database has the capacity to store all of those kinds of information. So I would imagine as it's grown already, it's probably going to start growing in terms of the different parameters that we collect in terms of those kind of inflammatory and cardiovascular risks. So we know that specific clonal hematopoiesis mutations in the general population are associated with the risk of cardiovascular and other events, not just leukemia. So whether that's true in these carriers is something that would certainly be important for their general health as well.
Okay, I think this will be the last question, and this is more of a general question in terms of the field of inherited predispositions. And the question was, did I hear you correctly that we've only solved the genetics of approximately 25% of MDS AML families?
I didn't say, no, I don't think I said that. I said I think we've only captured about 4% of the known RUNX1 families. I think there's data that's been published by our groups and other groups recently that if we look at the range of genes in the research sphere, so not necessarily present on clinical testing panels, we're probably at around 50%. So we can probably explain around 50% of inherited MDS AML with candidate gene mutations. So that tells you there's still a lot of discovery out there to be made as well.
Definitely. Well, great. I just want to say thank you again so much, Anna. This has been fantastic work, and thank you to all those who have collaborated with Anna and Hamish. It's just wonderful to see what we can glean from this data, and we hope that we can continue to add more to the database and learn more as we work together as a network and a collaborative community. So thank you again to everyone for joining. And Anna has kindly agreed to recording this presentation, so we will have it available. I had a few questions come in regarding that, so we can certainly share the recording, but please, I think some of this data is yet to be published, so please be thoughtful about that as well. With that, thank you and enjoy the rest of your evening or the rest of your day, depending on where you are. Thanks again. Take care.
Thank you.
Bye.
Bye, everyone.